All right, today we're cracking open the modern investigator's bag of tricks, the Genetic Detectives Toolkit. So, here's the question. How do doctors actually unmask an invisible killer? I mean, when some mysterious infection pops up, it's a total race against the clock. The culprit doesn't have a face, it doesn't leave traditional fingerprints. So where do you even start looking?
Well, they go after the one thing every culprit leaves behind. It's genetic code. You see, inside every single virus, every bacterium, there's a unique blueprint made of DNA or RNA. And modern science has built this incredible toolkit to crack that code and figure out who the bad guys are with pinpoint accuracy.
So let's start with the first tool in our kit, which is all about matching genetic fingerprints. This is the classic, the foundational method, and it's called nucleic acid hybridization. Okay, so how does it work? Think of it like a key that fits only one very specific lock. Scientists create this tiny little thing called a probe. It's a short piece of DNA or RNA that is designed to be a perfect match for a unique part of the pathogen's code. When this probe is introduced into a patient's sample and finds its other half, it latches on, or hybridizes. It's a perfect genetic handshake.
And you can see that genetic matching game happening right here. The probe is tagged with a fluorescent dye. See that little glow? It gets sent into this huge sea of genetic material from the sample, but it's super picky. It will only attach to its one true partner. And when it finds it, bam, it lights up, basically setting up a flare to say, hey, the suspect is right here.
Now, one of the most powerful versions of this technique is called FISH, and that stands for Fluorescence in Situ Hybridization. The in situ part is the real magic here. It literally means in its original place, and that allows scientists to see exactly where the culprits are hiding, right inside the patient's own cells. The whole process is pretty straightforward. First, you take the cells from a sample and you fix them onto a slide, basically locking them into place. Then, you introduce those fluorescent probes and let them go find their targets. After that, you do a quick wash to get rid of any probes that didn't find a match, and then it's time for the big reveal under the microscope. This diagram here, it shows that journey from sample to signal perfectly. You can literally see the probe making its way inside the bacterium, finding that specific bit of genetic code it's looking for, and making the whole organism just light up. It's a direct visual confirmation, no guesswork needed. And here it is, the final clue. This is what scientists are actually seeing when they look through the microscope. These are human chromosomes, and those little glowing dots are where the FISH probes have hit their mark. That visual evidence is just undeniable. It can confirm a pathogen is there, or even spot a genetic abnormality. What a payoff. So why is this so important in a real-world clinic? Well, with FISH, doctors can pinpoint pathogens right inside a patient's tissues. They can find sneaky bacteria hiding out in blood cultures, and crucially, they can even spot the specific genes that make a bug resistant to antibiotics. It gives them a visual map of the entire infection. All right, so that's like matching a single fingerprint. But what if you need more? That brings us to our next tool, reading the full code with DNA sequencing. If FISH is like matching one fingerprint, then sequencing, well, that's like finding the suspect's entire diary and reading it from cover to cover. So what is DNA sequencing, really? At its heart, it's simply the process of figuring out the exact order of those 4 chemical building blocks, the A's, T's, C's, and G's, in a microbe's genome. You get the whole, unabridged genetic blueprint of the organism. Now, this comes in 2 main flavors. You've got Sanger sequencing, that's the original. It's incredibly accurate, but it reads just one piece of DNA at a time. Think of it like perfectly proofreading a single page. And then you have the modern powerhouse, next generation sequencing, or NGS. This thing is like digitizing an entire library all at once, reading millions of DNA fragments at the same time. And this is basically how that powerhouse works. The whole workflow starts by pulling the DNA out of the sample. Then, in a step they call library preparation, which is just a fancy way of saying they chop the DNA into tiny, manageable pieces, it gets ready. A machine then reads the genetic sequence of all those little fragments simultaneously. And finally, some seriously powerful computers stitch all that data back together to give you the complete blueprint. And the applications here are just game-changing. Sequencing lets us identify completely new or super rare pathogens nobody's ever seen before. It lets epidemiologists track viral outbreaks like we saw with COVID-19 in real time, literally watching them evolve. And maybe most importantly, it can uncover the exact hidden mutations that turn a regular bug into a drug-resistant superbug.
It is the ultimate intelligence-gathering tool. All right, for our last tool, hold on, because we're jumping straight into what feels like science fiction. We're talking about the CRISPR revolution. This is the futuristic gadget in the toolkit that, just a few years ago, was purely theoretical, but now it's very, very real.
Now, you've probably heard of CRISPR for gene editing, right? Well, forget that for a second. In diagnostics, we use it totally differently. Here, the CRISPR-Cas system acts like a programmable search engine for genes. We tell it exactly what sequence to look for, it hunts through a sample with incredible accuracy, and when it finds it, it sends up a flare. Let's break it down. Step 1 is this search. You've got this enzyme, the Cas enzyme. That's the blue shape you see here. Think of it as the workhorse. It's paired with something called a guide RNA, which is basically its GPS coordinate. This guide leads the enzyme like a genetic bloodhound to the exact DNA or RNA sequence of the pathogen we're hunting for. And here is the really genius part. Step 2, the alert. Once that Cas enzyme finds and latches onto its target, it just goes nuts. It activates this sort of collateral damage mode, and it starts shredding not just the target, but also these nearby reporter molecules that were added to the mix. These reporters are designed to release a fluorescent signal when they get cut, so a successful search sets off a bright, glowing alarm. So why is everybody so excited about this? Well, first off, speed. CRISPR diagnostics can give you results in under an hour. Crazy, right? They're also extremely specific, which means you get way fewer false positives. All of this is paving the way for cheap, portable tests you can use anywhere. Not just a fancy lab, but a doctor's office or even a remote village. It's a total game changer for quickly detecting viruses like COVID-19. Okay, so we've looked at 3 amazing technologies. Let's pull back and see the whole toolkit together. Because the real art of being a genetic detective is knowing you have the right tool for the right job. This table really breaks it all down for us. You've got hybridization, like fish. Its core job is matching a sequence, and its key advantage is that visual localization. It's perfect for seeing where a pathogen is hanging out in tissue. Then there's DNA sequencing. Its function is reading the whole sequence, giving you that comprehensive detail you need for investigating outbreaks and total unknowns. And finally, CRISPR diagnostics. Their job is finding a sequence with unbelievable speed and specificity, which is just ideal for rapid point-of-care testing. So look, the big takeaway here is that no single tool is a magic bullet. A modern genetic detective doesn't just rely on one method. They use the whole toolkit, combining the power of visualization from fish, the deep genetic detail from sequencing, and the lightning-fast detection from CRISPR to create this unprecedented arsenal in the fight against disease. And that leaves us with this one incredible thought to chew on. As these tools get faster and smarter and more portable pretty much every single day, how is this going to change medicine forever?